a reminder, we are in the DNA replication, structure and replication chapter. So I was talking about, oops, hang on. I was talking about the kind of general DNA structure on Monday. And now we're going to go into some slightly more complicated structure, since DNA is actually two chains of nucleotides wound together. So I'll talk a little bit more about the structure, and then we'll get into the process of how DNA actually copies itself and makes new versions of itself.
I don't see this PowerPoint on campus.
Oh, is it not? Oh, dang it. Oh, sorry, guys. Hang on.
The last one's the genetics linking.
Okay. I will get it up there ASAP. Thank you for pointing that out. I don't know how I forget that sometimes. Oh well. All right. Yeah, always tell me if something's not up there. Yeah, okay, so it'll go in that same module. File. Okay, there it is. Okay, so both of them now, the PDF and the PowerPoint should be up there. Thank you for pointing that out. Maybe refresh if you don't see it just yet. All right, sorry about that. Let's go back to there. Okay. All righty. So the secondary structure, I talked about, like I said, the one nucleotide chain and kind of what a nucleotide is made of. Now we're going to get into how two of those come together and create this really lovely double helix that is DNA. Okay, so you have this one, so these are two chains essentially of nucleotides. Remember, the nucleotides have a phosphate, a sugar, and then a nitrogenous base. So it has those three parts. So this would be one nucleotide on one strand, one of the polymers. This would be the nucleotide on the other strand. So it's a complicated single strand, and then when you add them both together, it gets even more complicated. So here you have one nucleotide, for those of you on Zoom, phosphate, sugar, nitrogenous base. On the other side, nitrogenous base, sugar, phosphate. So you have two of those attached by hydrogen bonds. So I talked about what bonds the individual strands together. So the nucleotides on one of these strands is bound together through phosphodiester bonds. So you want to know that name. So each of these, if we're talking up and down, those are phosphodiester bonds holding those together. Between the two strands, so side to side, these are hydrogen bonds. Those are the only two bonds you need to know, or those are the only two bonds really, yeah, that you'll need to know for DNA, phosphodiester and hydrogen, and know what they're actually bonding together. Another term that's important is anti-parallel, which just means the two strands, so here the left and the right, are basically oriented in opposite directions. And what I mean by opposite directions is, if you remember, I mentioned the 3' end and the 5' end idea. This will come up a lot again today. So the 5' end has, you don't have to be able to identify it. I'll tell you what the 5' end is, but the 5' end of any strand of nucleotides is going to be where there's a phosphate sticking off. The 3' end will be by default the other side where there's an OH sticking off. So here you can see the 5' end of this strand is at the bottom on the right. The 5' end of the strand on the left is at the top. So they're anti-parallel. Yes,
You said on the 3' end, what sticks out?
the OH.
Okay.
Yeah, so the OH defines that 3' end. I should have written it on here, sorry. Okay, so the ends are opposite of each other. That's the anti-parallel nature. That'll come into play in a big way when we talk about how it actually replicates itself. Okay, more about the nitrogenous bases, which is, I think I mentioned this when I went through the three parts that make up a nucleotide. I said the nitrogenous bases and I bolded them. I bolded that word on that slide and I said, this is what we really care about when we're talking about this DNA replication process. The sugar and the phosphate, yes, obviously it's necessary, but it doesn't really, it's not the code for the DNA. The nitrogenous bases are the codes for the DNA. So you have 4 of them. So you have those 4 letters, A, G, T, and C. Adenine, guanine, thymine, and cytosine. You want to commit those to memory. They are broken down into 2 different categories. So there are kind of 2 different molecule types, so large categories. You have the pyrimidines down here. Do I have the, yeah, it'll come up in a minute. So I don't know why I have that animated. We'll put both of them up here. You have purines and you have pyrimidines. The purines are adenine and guanine. So they have 2 rings. So you'll see the 2 rings here. Those are purines. An easy way to remember that adenine and guanine are the purines is this little, I guess it's a mnemonic device, pure as gold, the A and the G. Adenine and guanine are both purines, so A and G here. You want to know that those are the double ringed nitrogenous bases, meaning they have 2 rings of carbons stuck together. You'll notice the others only have 1 ring, so those are single rings. That's the main difference between these 2 groups. So this other group are known as the pyrimidines. Single ring structure, this is going to be cytosine and thymine. And an easy way to remember that they are in the same category is that they have a Y in their names, just like pyrimidine has a Y. So thymine and cytosine both have a Y, pyrimidine has a Y, so those are in the same category. So yeah, pyrimidine. And there are no Ys, obviously, in adenine and guanine. So pure as gold, and then the Y will hopefully help you keep those 2 groups separate. However, the same groups don't bond with each other. So purines don't bond with purines. Here we're talking across 2 strands. So imagine these are 2 DNA strands here. A purine is going to bond with a pyrimidine every time. So they bond with something that's not like themselves. They want something different, I guess. So bonding between strands occurs only between purines and pyrimidines. And specifically in this way. Adenine bonds with thymine, cytosine bonds with guanine. So that gets a little more confusing because you have to know which are the purines and which are the pyrimidines, but then know the 2 that actually bond with each other, the pairing up of the A and the T and the C and the G. And these are the only, this is the only way these will bond. A is not going to bond with G. It's always A plus T or C plus G. So if you know one half of the strand, you can predict what the other half is going to look like because you know exactly what those nitrogenous bases have to bond with. And that's essentially how DNA is built. just matching up the nitrogenous bases with the correct opposite part of the pair. All right, so those are the purines, pyrimidines, and then the bonding pattern. And then, so I have a list here of sort of what I've been through with the DNA structure, kind of a review of the really important aspects of what makes DNA unique and special. So DNA is a polymer of nucleotides. So nucleotides are the monomer, a bunch of those strung together basically makes DNA. So you want to know the term nucleotide. It is actually two of these nucleotide strands wound together into that helix shape. So it's a double-stranded helix, that's what we call it. What is a nucleotide? It is made up of three parts. It's, like I said, a sugar, a phosphate, and a nitrogenous base. The nitrogenous base is either a purine or a pyrimidine. The phosphate's always the same, it's just a phosphate group. And the sugar is always the same, it's deoxyribose. That's the name of the sugar. The only thing that changes is the nitrogenous base from one nucleotide to the next. And the base pairing. So base pairing, I mean which two nitrogenous bases actually bond to create that double-stranded helix. Purine plus a pyrimidine. So that's going to be A plus T, C plus G. Those are the bonding between the two strands. That's the bonding pattern between the two strands. Those are held together through hydrogen bonds. So the adenine bonds with the thymine through a hydrogen bond, same with the cytosine and the guanine. What else do I have? Ah, yes. The strands, the two strands, when you have the double helix, they're arranged anti-parallel to each other, which means if we name the ends 5' and 3', the 5' end of one strand will match up with the 3' end of the other strand. So they're going in the opposite direction. So anti-parallel is a really important term that you need to know. Is that it? Yes. Okay. Any questions about this? The basic structure, kind of a review of what DNA actually is before we get into how new DNA is made. All right. So the discovery of DNA as a double helix was a huge, huge deal. People didn't know actually, to begin with, what was the genetic material, like what passed on our traits. They thought it was proteins for a long time, because proteins are obviously really important. But then they eventually discovered, no, it's DNA, and then they tried to figure out the structure. So these four people, you'll probably, if you ever hear about DNA discovery, you'll hear about these first two mostly, Watson and Crick. They're the ones who discovered how this was actually put together, that the nitrogenous bases were on the inside, holding these two strands together in a double helix. Rosalind Franklin and Maurice Wilkins also contributed to it through some other means. It was a lot of kind of collaboration. And I don't really, we have so much to go over today. I was going to show you this video. If we have time on Friday, I'll show it to you. Or you can watch it on your own if it's of interest to you. But hearing how this double helix was, the whole process of discovering the double helix and people getting mad at each other and drama happening and all kinds of stuff in the scientific community, it's pretty interesting. It was kind of like just this huge mystery that was figured out. And then let's see, Watson, Crick, and Wilkins, they won a Nobel Prize for discovering this. And Rosalind Franklin had already died by that point, so she was not awarded the Nobel Prize, unfortunately. But it's an interesting story. Like I said, if I have time, I'll show it to you, but the link is here. If you want to watch it, feel free. But for this slide, just know these 4 names. These are the 4 people who are responsible for discovering the double helix, so the structure of DNA. So yeah, like I said, Mendel had no idea about any of this. When he was doing all of his pea plant experiments, he didn't know what was going on. He just kind of looked at the patterns of inheritance. So this was far, far after Mendel did all of his experiments. This was in the 20th century. Okay. So getting into the replication process. Chromosomes, as a reminder, if you think back to the cell cycle, that big pie chart that I showed you when we went over mitosis, chromosomes are replicated, duplicated during the S phase of interphase. If you don't really remember when that is, go back and look at those slides, look at your notes on the cell cycle. You definitely want to know the S phase of interphase is where everything I'm about to tell you is happening. So it's before meiosis happens. The chromosomes have to be duplicated in order to actually create new cells. So the whole point of this is to create new cells that also have DNA in them. That's why we're duplicating the DNA. So when a chromosome is duplicated, that's the same thing as DNA replication. Chromosomes are just condensed forms of DNA. All right. So as an overview, I know this looks like a lot, so just bear with me for a second. I want to point out some important aspects because in future slides, I'm going to be zoomed in a lot more on like one part of a DNA strand. So I kind of want to point out a few of these terms and talk about generally what's happening. And then I'll go into more detail on, all of these terms. Okay, so in general, so up here we have the parental DNA, that's the blue colored DNA. We'll imagine those are just 2 strands of DNA. Yes, they're normally wound into double helix, but for simplicity's sake, we're just drawing them as parallel. So we have the parental DNA, that is what's going to get replicated. At the end, we'll have 4 DNA strands basically, so 2 double helices. Each of the parental strands, the double helix is basically separated, like you can see here, kind of in segments, separated, and then new DNA, new nucleotides are put down to match up with the parental DNA. So you'll end up with a new strand of DNA that was created during this process, matching up with one of the original strands. So you have a mix of old and new. So for you guys on Zoom, up here we have the parental DNA the double helix will open up and then new nucleotides will be laid down to create a new strand of DNA. So in the end, you have parental DNA and then new DNA. So the basically newly formed DNA then attached to the parental DNA. And then that winds up into a double helix again, but it's not shown here. Okay. So this is happening in both directions, as you can see. That's going to be a big part of what I'm going to have to explain to you, kind of how that works. But essentially, the DNA is unzipped, and then both of those original strands are copied. So you put new nucleotides down, and then you have those will separate and then become what you have down here. So that's the overall take home. I'm going to walk you through it pretty slowly, step by step. There's a lot of steps. It's, as you might guess, like most of biology, a complicated process. So we're going to walk through how this is done, and then the proteins that are required to make this happen. So the tools as well as the whole process. Okay. So first I'm going to go through some terminology that's used to describe the DNA replication process. So like I said, the DNA helix is split open, right? So if you imagine a double-stranded molecule, it gets split open. So you have the two parental strands, and here you can see those nitrogenous bases, the letters for the nitrogenous bases that we talked about, the C, the T, the A, and the G. So that'll be opened up. And then a copy is made of each parental strand. So what I mean by a copy being made is that the correct pairing of nitrogenous bases is added. So new nitrogenous bases are put down that will match up with the original parent strand. So here you have a C at the very beginning. That means the new strand, there's going to be a G put down because that's the pairing pattern, right? C plus G. Next, you have a T. So that's going to match up with an A. An adenine, will be brought in and put in that second spot to match up with the T. So for you guys on Zoom, if you couldn't see that, here we have a C on the parent strand. This daughter strand is the new growing strand. So a G is put down to match up with that C. C plus G and A plus T. Those are the base pairing patterns. Here we have a T in the second spot. So that means an A has to come in to match up with the T and so on. And then you just continue on down the line until you have, well here, this is showing both parent strands basically splitting apart and the new daughter strands growing in the middle here. That whole process, the term for that is called semi-conservative because one of the parent strands is conserved. So semi-conservative just means that you end up with your two new double helices of DNA having one parental strand and one daughter strand in each. It's not like you have two parent strands, like the two old ones and the two new ones stuck together. You have half old and half new in each of the new double helices. That's semi-conservative. So that's the first characteristic I want to talk about. Oh yes, this might be a better image for you. I kind of like this diagram. So it's a little bit easier to read. You have the parent DNA where you have the two strands here and here. You can see they're matched up A, T, C, G. They're split apart or unzipped. So here you have the parent, those are unzipped. And then you have the new bases laid down to match up with the originals. So that's the semi-conservative. You have a daughter strand and a parent strand. Another term, or I guess characteristic, is that DNA replication is going to begin at what's called the origin of replication. That's pretty easy to remember. It's basically just a specific sequence of nucleotides. You don't really need to worry about what that is, but that specific sequence of nucleotides will cause or indicate to the proteins that are being used to open up that double helix. Anything else on here? No. Okay. So replication of origin of replication is just where the whole process starts, kind of bubbles out. That's a pretty straightforward term. Okay, this next aspect is probably one of the most confusing parts of understanding DNA replication. We say that DNA replication is bidirectional. So what that means is that it's happening in opposite directions if you're talking about one DNA molecule that gets unzipped. So on one half of that DNA, you have continuous synthesis. On the other half, it actually happens in individual segments, and I'll talk about why that is, but that's the bidirectional aspect. So on this strand here, the new DNA strand, which is going to be this darker purple, or on Zoom, it's pink. So this pink strand is the new strand being built. It just keeps going and it's going towards the right, towards the replication fork. I'll talk about that in a minute. The other strand is being built in the opposite direction, away from the replication fork, and in fragments, which I'll talk about those too. So here you see the directions are opposite. Okay, why is this happening? Because remember, the DNA strands, if we're talking about the original just parental DNA helix, they're anti-parallel, right? So the 5' end and the 3' end are on opposite ends of each of those strands. I bolded this because you want to definitely commit this to memory, and if you can remember this, this whole process will make a lot more sense. New bases, which I mean the new nitrogenous bases, the A, the T, the C, the G. Those are always added to the 3' end of the new strand. So I'm going to talk about this, what's called the leading strand on top first. I think I might have this. So hang on. Okay, yeah. So here, this original parent strand, here's the 3' end, so that's marked as 3'. That means the new strand, we have to start with the 5' end, right? Because they're going to be anti-parallel. They have to be anti-parallel. The parent strands originally, and then when the new strands are added on, they're also anti-parallel. So the 5' end of the new strand is here. We can't add new bases to that side. So they get added towards the other end, which is the 3' end. So they're getting added in this direction. Because the other parent strand on the bottom, its 5' end is down here. That means the 3' end of the new strand is off to the left here. So that means that strand is growing towards the left on this diagram. So you're going to get really tired of hearing me say 5' and 3' over and over and over again. Just remember, new bases, when we're talking about the new strand of DNA that's being built, they can only be added to the 3' end. So if you can find the 3' end, which I'll always label it for you, you can figure out which direction that new strand has to grow in. Okay, so that's the bidirectional aspect. One is growing to the right, one is growing to the left. So what about these fragments, basically, that are on that bottom strand? So you have the top strand here is labeled the leading strand. That's a term you want to know. The leading strand is the one that is growing towards what we call the replication fork. So you can imagine the DNA kind of continuously being unzipped in this direction. Where that last part of the double helix is still intact, that's called the replication fork. So it'll continuously kind of move this way as more and more DNA is being copied. So the leading strand grows continuously towards the replication fork. It's pretty straightforward because as this gets unzipped further, it just exposes new DNA that can now be copied. So it can just keep going, no problem. That's the 3' end, so it can just keep on adding bases. The other strand, that's the easy one. The other strand is called the lagging strand. And it's actually formed in fragments that are called Okazaki fragments. You want to know that term? Okazaki is the guy who discovered them and figured out how this worked. That strand, as we said before on the previous slide, is growing away from the replication fork. So it can't grow in this direction because the 5' end is here. We can't add new bases in the direction that the DNA is actually unzipping, which causes a problem. So we'll start with this segment here, and I'll try to describe what is happening if you haven't already figured it out. So for you guys on Zoom, this leftmost fragment, we'll imagine that's the first one that gets made. So you can imagine the DNA, the replication fork, was here at one point, right? It wasn't unzipped any further than this. So this is where the DNA replication started for the lagging strand. It has to grow in the 3' direction. So you have to add new bases going away from the way that the DNA is actually unzipping and exposing more DNA to be copied. So the growth and the unzipping are going in opposite directions for the lagging strand. So
Is that a, like, is that a form of the anti-parallel?
Yeah, it's because of the anti-parallel aspect of DNA. Yep, exactly. If it weren't anti-parallel, they could just both grow in this direction. So we're adding new bases towards that side. So towards this 3' on the lagging strand for you guys on Zoom. And then, so if we were only unzipped to here and that got laid down, now we'll imagine that the DNA is unzipping further and further back, right? Exposing more of the parental DNA to be copied. So now the process kind of has to jump back and make up for that new DNA that just got exposed. So it continuously does this jumping, but it still has to put new bases down in this direction, away from the replication fork, so to the left on this diagram. So for you guys on Zoom, this was the point at which it was unzipped to lay down this first segment. Now we'll imagine the DNA unzips a little further back, so say to here. Now you have all this new DNA that's opened up to be copied, basically. So now this new segment can be laid down, but you still have to add bases towards the 3' end. So it just keeps jumping back as more and more DNA gets exposed. And it's hard to understand on a static picture like this. I have, after we go through the whole process, I have an Amoeba Sisters video that will hopefully kind of help explain that in actual kind of video animation. Okay, questions about that? If you're totally confused, it's okay. We'll do some more work on this. I have a worksheet for it, depending on how far we get today. I'll either let you all do it before Friday or we'll work on it together on Friday. Okay, so we have the semi-discontinuous aspect. So it's continuous on the top, well, in this case, the top strand, discontinuous on the bottom because you have these fragments. That's the semi-discontinuous aspect of it. And I think the most difficult to understand. All right. Whew. So now I'm going to talk about some of the tools that are needed for DNA replication. There's a bunch of proteins, which are these are all enzymes basically, but proteins that do all of this. It doesn't just magically happen. Proteins have to be used to, well, I'll tell you what each one is used for. So helicase is one of the first ones that comes into play. It's going to unwind the DNA at the replication fork. So you have it wound up into this double helix. Helicase comes along and just unzips it. When that DNA gets unzipped, it's fairly unstable and wants to rewind right away. It wants to come back together. So single-stranded binding protein, or SSB protein, basically stabilizes the unwound DNA and keeps it from rewinding up. So helicase unwinds, the SSB protein keeps those two strands separated. And then at the other end, so the two strands want to come back together. SSB keeps them separated. At the other end, if you imagine like two hoses kind of coiled together, sort of in a double helix, if you pull them apart and unzip them, it just gets really tangled up and compacted ahead of where you're pulling them apart, right? So the topoisomerase basically keeps the DNA from what we call doing this supercoiling aspect, so just becoming more and more tightly wound ahead of the replication fork. So there's a lot of these are involved in just getting the whole DNA double helix ready and stable. Helicase unzips it, SSB protein keeps it from rezipping, topoisomerase keeps it from supercoiling at the other end and being compacted. And then before any DNA can be laid down, there's something called an RNA primer that has to be put down. So to complicate things even more, you can't just start laying down the DNA nucleotides. You have to put down some RNA first. We haven't talked about what RNA is, but just take that for what it is right now. It's just an RNA primer, kind of sets it up, and then from there, you can put down new DNA nucleotides. So primase is what does this. Primase lays down these first few RNA, this little section of RNA, basically, at the very beginning of the new strand of DNA. So primase creates the primer. And DNA polymerase, if you don't remember any other, which you should still, I'll ask you about the other proteins. You want to know DNA polymerase. This is the one that puts down all the new nucleotides. So it's what's matching those complementary bases. If there's an A, it's going to add a T. If there's a C, it's going to add a G, and so on. DNA polymerase is what's making the new strand of DNA. And then because we can't leave these little sections of RNA primer in there, it wipes those out and puts down DNA in the place of the RNA primer as well. So it does a lot. It's kind of the workhorse of the whole DNA replication process. But they're all important. So DNA polymerase, yeah, forms new strands. I like this diagram. It's just very simple. This is what's forming the new strands of DNA. And then finally, we have ligase. That's sort of the glue. It's joining, all those Okazaki fragments are not connected when they're made. So they have to be glued together. So the ligase is what's joining those together. Okay, so those are the 6 proteins that you need to know. That's a lot. But in the review video for the Amoeba Sisters DNA replication process, they go through each of these and kind of describe what they do again. So that'll be a good review. All right. So now we know kind of generally what's going on, and we know the proteins that are involved. So now let's talk about some detail of the process. I've already kind of talked about some of the process, but we're going to go through each of these sort of main steps of the replication process. So there's the initiation, which is kind of the preparation for DNA synthesis. Elongation, where the DNA is actually being made, and then termination. So it has to stop at some point, and there's certain things that have to happen to make the process stop. All right. And you'll see these names again, initiation, elongation, and termination, when we talk about sort of the next 2 steps in that central dogma of biology, so transcription and translation. They all have the same general steps. Okay, so I'm going to go through each of these on separate slides. What happens during initiation, what proteins are involved, same for elongation, and then termination. Okay. So initiation, first we have to separate the original strands, right? We got to unzip it because we have to copy each one. So DNA strands are unzipped by helicase. Here's helicase, it's unzipping in that direction. Do I have this later? Well, topoisomerase is over here keeping it from supercoiling. And then single-stranded binding proteins, that SSB protein, is essentially keeping that double helix separated. So we have those three proteins, which I have listed later on here, involved in the initiation process. So the single-stranded regions are stabilized by the single-stranded binding protein, aptly named. And then we also have, this is still the initiation phase, so we're laying down this RNA primer. RNA is slightly different from DNA. You don't need to worry about it right now. But know that there's an RNA primer that's laid down by primase. This will be the starting point for DNA, for the DNA strand being created. Okay, so here's a list. I mean, they're on the diagram, but here's a list of the proteins that are needed for initiation. So I did this for each of the main steps, just so you understand which proteins are needed, and you should know what they're doing, obviously, which we just kind of went over. So we need helicase, SSB protein, primase, topoisomerase. There's a lot of proteins are involved in just getting this thing started. All right, so now we have this little RNA primer, which would have been laid down here too, but we're just gonna focus on this aspect. So now the DNA in the elongation phase, we're gonna add new DNA bases in that direction off of the 3' end of that RNA primer. So the elongation phase is where the DNA polymer, so the many nucleotides strung together to make a new strand of DNA is being synthesized. Some reminders. This involves complementary base pairing, A with T, C with G. If there is an A, the new strand has a T on it. If there's a C, the new strand has a G on it, and vice versa. And this is only happening in the 5' to 3' direction, which means the new bases are added to the 3' end of the growing strand. The new strand, the daughter strand, whatever you want to call it. And again, it's bolded. You definitely want to know this. The RNA primer is removed and replaced with DNA. We don't want little segments of RNA in our DNA, so we got to take that primer out, put in DNA nucleotides. The only two proteins that are needed here, again, DNA polymerase, it's doing most of the work. It's adding the new nucleotides to the daughter strand. And it's replacing that RNA at the beginning, that little chunk of RNA, with DNA. And as this is happening, any fragments, like the Okazaki fragments, are being glued together by ligase. So this is, oh yeah, sorry, yes.
I wanted to ask, what does it mean when they synthesize?
When they synthesize, that just means adding the new nucleotides. So let me go back to an image. So if this is, here, I'll go back for you guys on Zoom too. So here, this is kind of representing an individual nucleotide. So you can think of it as like an A or a T or a C or a G. Synthesizing is where a new one is added to the other side. So you're adding new nucleotides to the growing strand. Actually, I think, yeah. Should have gone the other way to the next slide. So synthesis would be, this is the new strand that's growing, this kind of teal colored thing. So new nucleotides that are being added, that's synthesis. So here we have, I have a slide here for the leading strand, which is the one that just keeps going, no problem. And then on the next slide, I'll show you the lagging strand, which is a little bit more complicated. Okay, so leading strand, the DNA is being unzipped to the left here, so to your left, by helicase. And then DNA polymerase is coming in and putting down these new nucleotides. As the DNA unzips towards the left, when it unzips a little further, it can keep doing the same thing because that's the 3' side of the new strand, so no problem. Just adding new nucleotides as the DNA unzips further and further. So yeah, down here, this is further unzipped. Okay. The lagging strand, I have a slightly different looking diagram here, just kind of simplified. So we talked about the leading strand. The leading strand isn't always on top, I should say that. It just depends. I think for most of these diagrams, to simplify things, they keep it on top, but top versus bottom doesn't matter. So the lagging strand here on the bottom, the DNA is unzipping this way, right, to the left still. So we're still unzipping the DNA to the left. There's a replication fork. Unfortunately, on the lagging strand, the 3' end of the lagging strand is going this way. So we have to add new bases. We're growing the strand, synthesizing the strand away from the replication fork. So we have to create basically a fragment here if the DNA were only unzipped to here, and then as it unzips further, you just create fragments kind of working towards the replication fork. So each of those are called Okazaki fragments, and they're not glued together yet. That's where the ligase comes in. They have to be joined. So yeah, for you guys on Zoom, we got to lay down a fragment because we have to grow the DNA strand in the 3' direction and then jump back as more DNA is exposed, and then jump back and kind of continuously kind of do this thing. I don't know how to describe that in words, but it's unzipping that way, but growing in this direction. I think that's the main aspect of elongation. Questions about the Okazaki fragments? Okay, I had a really hard time understanding this when I learned it the first time. Maybe, I don't know, maybe I just wasn't listening that closely. But hopefully it's making sense to you guys. This is just sort of a more complicated version. Another aspect of the Okazaki fragments is that for each one, just like on the leading strand, you have to lay down some RNA first. So some RNA primer, that's the red here. RNA primer gets laid down and then DNA grows off of it. So there's a lot more work involved with getting the lagging strand copied compared to the leading strand. All right, and I'm going to do termination super quick so we at least get through. Termination's pretty simple. Nuclease I didn't talk about, but it's going to remove any errors in the DNA. So it kind of proofreads itself. Sometimes the wrong base is laid down, the wrong nucleotide. So nuclease will fix that. DNA polymerase will come in, add the new bases that need to replace any that are kind of messed up. And then ligase will glue all that together. And then that wraps up the whole process of DNA replication. So I'm going to post, and I'll email you guys, I'll post a worksheet that can be really helpful in applying some of these concepts. It won't be due, but we'll go over it on Friday, sort of as part of the review for the exam. So the exam will still be Monday. we'll finish up a few more aspects of this and then do a review on Friday. I think that's it. Okay. And we'll watch the Amoeba Sisters video also on Friday.